The next presentation is going to be given by Jakob Schreiber from the University of Washington, and the presentation is going to be entitled "A Pitfall for Machine Learning Methods Aiming to Predict Across Cell Types."
Thank you. All right, well, thank you for the introduction. I'm not actually Dr. Schreiber just yet. I'm in my last year of my PhD with Bill Noble at the University of Washington. You may have heard his keynotes earlier in the conference, and if you have a particularly keen memory, you may remember me as the person that he outed in his main keynote as liking Gossip Girl. Unfortunately, Bill vetoed my presentation that was going to be primarily on Gossip Girl, thinking that wasn't really so bio-focused. And so we're going to be talking a little bit more about my research. In general, I'm going to be talking about machine learning for genomics. And more specifically, I'm going to talk about a pitfall you can encounter if you aren't careful with how you set up these problems. So when I say machine learning and genomics, I mean problems that kind of fall into this format. The idea is that you have some form of like genomic or epigenomic inputs to your model, and you'd like to make some prediction about genomics or epigenomics. So for example, you might try to use nucleotide sequence and some epigenomic measurements to predict gene expression. You might use those same things to try to predict transcription factor binding. That's been hot in the last few years. You might try throwing the kitchen sink to try to predict chromatin conformation, things like TAD boundaries, promoter-enhancer interactions, entire high-C contact maps. So these are the types of problems I'm going to be talking about, and in order to illustrate the settings and the pitfall, I'm going to focus on one, which is just using nucleotide sequence and various epigenetic measurements in order to predict gene expression. I want to be clear, though, that what I'm talking about is more general than just this problem. It's any of the problems here. I'm just choosing one in order to demonstrate. And so I'm also using data from the Roadmap Consortium. I'm using their gene expression data and the histomodification and DNase. So let's say that my goal is to try to predict gene expression. The first setting, an incredibly common setting in our field, is called the cross-chromosomal setting. In this case, you're typically trying to make predictions in the same cell type that you are training your machine learning model in. You do this by partitioning your loci, in this case genes, into your training set and your test set. Commonly, you'll take some chromosomes, put them in your training set, take other chromosomes, put them in your test set, and that's why it's called the cross-chromosomal setting. So let's say that I build some neural network models, and what I have is on the I can't aim this, but on the y-axis, what you have is accuracy. On the x-axis, what you have is complexity of the model in terms of number of parameters. So if my input are just epigenomic modifications, what you can see is that it seems like the performance improves slightly as you add complexity to the model, and then it kind of flatlines. And this makes sense. There isn't some incredibly complex regulatory code that encodes gene expression. Next, let's say that I build models using only nucleotide sequence. What you get is models that perform better than random chance, but worse than if you used epigenomic modifications. And hello. Oh. Awesome. Okay. So let's say that I'm building some models using nucleotide sequence. And these are a combination of just dense neural networks and convolutional neural networks all mixed together. What you get is that you get performance that's better than random chance, but worse than using epigenomic modifications. And this is something that's been seen in the literature quite a bit, that there are some nucleotide motifs that indicate if a gene is going to be expressed or not. And then as you make the model more complex, it seems like you get slightly worse performance, indicating you might be overfitting. In this case, we also kind of smush nucleotide sequence and epigenetics together. You get kind of an improvement over nucleotide sequence. We didn't spend too much time trying to optimize it in this case. So one of the main points I want to make in this presentation is that the primary reason you would ever want to do cross-chromosomal work in this cross-chromosomal setting is when you build a model and then you use it to understand better biology. There have been a lot of great talks, including that BPNet talk that happened a few days ago, where what they did is they would train a model, and then they would interpret it. And by interpreting it, they learned something about biology. If all you are doing is building a model and saying my accuracy is higher than someone else's accuracy, I would say that that is useless. It doesn't matter, because there is no real evaluation setting where someone is going to hand you half of a genome and say, we need to make that prediction. And so I would avoid using this setting if your goal is simply to build a better predictive model. However, if your goal is to build a better predictive model, I would recommend this setting, which is the cross-cell type setting. This is a much more realistic setting where what you are doing is you are taking data from some cell types and you are making predictions in a new cell type. This is common when the thing that you're trying to make predictions for are very expensive to measure. Like, for example, getting a whole high-C map. Maybe you want to make predictions for high-resolution, high-C maps in the, I don't know, dozen or fewer that we have, so that we can now make predictions in all of the other cell types. So, yes, this would be a much better setting in order to compare models. And so if you are writing a paper and saying, my model performs better than some other model in this setting, I would say that that is a strong contribution. So let's say that I'm going to be building these gene expression models. If I use only epigenetic inputs like last time, I see basically the same thing. I'm getting roughly the same accuracy as before, and it's kind of flatlining. Now, I use only nucleotide sequence. And what's interesting is that as the complexity of my model increases, so does the accuracy up until a certain point. These models get very large, that if you have 10 to the 5th parameters and you're still increasing in the accuracy, it seems like maybe you're getting some form of super complex motif that's really indicative of gene expression. I hope I can see some skepticism on your faces from this type of result, but this is something you can try out at home really easily. And so you get a happy face because the results initially make you happy. And then when you add in epigenetics, you get basically the same thing, where it looks like adding in epigenetics makes things slightly better as well. So a third setting you could use is called this hybrid setting, because it is a hybrid of the cross-chromosomal and the cross-cell type setting. In this setting, your test set are loci that you didn't see in cell types that you didn't see. And so your model has to generalize across both cell types and assays. I think there's a little bit of debate about how useful this setting is, but I think that it's definitely useful to evaluate whether or not your model is overfitting to particular trends. So in this case, what I'm going to do is I'm going to use the exact same models that were trained in the cross-chromosomal setting, the cross-cell type setting that I just showed you, where you saw the increase in performance, and we're going to evaluate them on loci that they didn't see before. Again, if we use epigenetics, we get the same boring result. That seems to flatline around 0.9. Now, let's use these sequence models. These are the exact same models that were trained we saw increasing performance on. On new loci, we get poor performance. We see performance similar to the cross-chromosomal setting. How can that be? Why is it that these models that seem like they were doing incredibly well in the cross-cell type setting are now performing very poorly in this hybrid setting? Well, in order to figure this out, it's helpful to kind of look at the real data. We have epigenetics in the middle there. If we look at the gene expression values, you might get a table that looks something like this, where the dark green are the highly expressed genes and the light green are the lowly expressed genes. What you'll notice is that this is fairly conserved, it's fairly similar from one cell type to the next. If you take the average expression across all of the cell types in your training set, it seems like it would be a pretty good predictor of some new test cell type. Here, you'll notice that in this new test cell type that wasn't in our training set, that we have a lot of similarities with just the average. Unfortunately, if we just take the average, while it seems like it may be a good predictor, it's missing all of the interesting, useful biology. The useful biology is the stuff that differs from one cell type to the next. Your average activity isn't going to get any of that, but it's going to be the most biologically interesting thing to study. So gene expression, to be clear, is not the only setting in which this is the case. Let's look at this histone modification just mapped across some region in chromosome 1. These are 6 very different cell types from each other, and the activity is pretty similar. You have these regions like this one, which are never active. You have many peaks that are active in all of them, but you have some variable peaks, and these variable peaks are likely the most biologically interesting ones. But if you're building a machine learning model, you probably want to show that you're doing better than just this average. CTCF is a protein that's very similar from one cell type to the next. You can see how similar it is here. And looking at just mRNA experiments, that there are vast regions where nothing is happening. So this similarity suggests some mechanism behind what's going on here. We're back to the cross-cell setting here. These are the same results that I was showing you before. Let's take that average activity that we just found. Let's take the average gene expression across all of the training cell types and plot the performance of that. Well, it's right there. That it looks like as we're increasing the complexity of our models, we are approaching but not actually beating using just this average activity from all of the training sets. That's because many phenomena are very similar from one cell type to the next. Now, we investigated this a little bit more. We said, maybe it's not so much the input exactly that's going on. Maybe we can perturb it and we can get rid of this. That would show that nucleotide sequence might not actually encode real motifs. So what we did is we took the nucleotide sequence and we shuffled it. Same composition of nucleotides, just shuffled from before. And that's the magenta here. We get basically the same performance as when we use nucleotide sequence by itself, but we've unambiguously removed any real biological signal that might be there. We then went a step further and said, what if instead of using nucleotide, one hot encoded nucleotide sequence at all, what if we just use random numbers? That's the cyan. We get basically the exact same result if we just use random numbers here. So what's going on? Well, in order to investigate this, we decided to inspect these neural network models. This is kind of a canonical convolutional model. All of our models kind of follow this format. Well, what we have here at the end is some final hidden layer before we make our prediction. So if we focus on this final layer, we can look at what is the representation of all the loci in our model right before we make this prediction. And so what we see, what we've done here is that we're looking at 4 models that range from poor performance on the left-hand side to great performance on the right-hand side. Each one of the dots corresponds to one of the genes in our set, and they're colored by the average activity, where white means low average activity and kind of dark red/black means high average activity. What you can see is that as the model gets better at making predictions, allegedly, it is better organizing these genes by their average activity. All it's doing is memorizing the average activity. And I want to stop here and emphasize this. What is happening is that if you make models any model of sufficient complexity can take an input that is invariant across cell types, such as nucleotide sequence, and use that to memorize the average activity at that location. These models don't care what the actual sequence at a gene is. What they care about is the ability, what they care about is whether or not the model is complex enough to learn a mapping between that sequence and the average activity at that location. If you are including features that are agnostic to the cell type in your model, you have to make sure that your model is not simply memorizing this, because it's going to look like, as you add complexity to your model, whether or not it's more parameters or more complex things like attention or embeddings or whatever, that it's going to look like you're doing better when all you're doing is memorizing this better. So how bad can this get? Well, I put animations in going the wrong way here, so that's as bad as it can get. So these are results from a recent Encode Dream Challenge that was hosted. That the goal was to try to predict transcription factor binding using nucleotide sequence, accessibility, and gene expression. Here are the top 4 participants in various pastel colors, and then if we use the average activity across the training set for CTCF, it beats all of them. So this is a very prevalent, maybe it doesn't beat DX-Wong. I think that it kind of does due to the sparsity of the number of cell types that we have, but it's at least a very strong competitor here. To be clear, this is not building a model at all. There's no machine learning going on with the yellow. This is simply using the average activity at each locus in the training set as your predictor in the test set. You do incredibly well and can beat what might be state-of-the-art models as of last year. So, how can we make sure that we're avoiding this? One of the ways that we can do this is that we can partition our loci by the frequency which would say are active in the training set. What we have here is that on the x-axis, we have the number of cell types that a certain gene is active in, and on the y-axis, we have performance. If we take the exact same results I was showing you before, and I was just calculating the average precision over all of them, but partition them in this way, the models that use epigenomics alone outperform the models that use sequence alone. The models that use sequence alone basically are just at baseline performance because they're just, what we're doing is we're factoring out the average activity, and we're saying how much better do you do when you factor that out. And so, of course, the epigenetics, once again, performs better. So let me just wrap up. The first is that if you are using the cross-chromosomal setting, the best application of this is to train a model and then inspect it in order to learn new biology. If you then take my advice and move over to the cross-cell type setting, you need to avoid this pitfall, which is that if you are using the same loci in the training and the testing training set and the testing set, you need to make sure you're not just memorizing the average activity. And you can tell this is the pitfall because I've highlighted in red here. So I don't think that simply moving to the hybrid evaluation setting is the best thing to do, because ultimately your model needs to make predictions at every genomic loci, including the ones that it was trained on. Simply restricting it to a different portion of the genome isn't really solving the issue, it's just kind of sidestepping the issue. And so kind of more of a general thing is that I think that the fact that the average activity baseline is so strong and that it's so difficult for us to beat it indicates that there's a lot more knowledge we need to glean from the genome. Because if H3K4ME3 was always indicative of active gene expression, then we would always beat the average activity baseline. The fact that we don't indicates that biology is complicated. And as a computer scientist, I don't like that. So, I'd like to thank the co-authors of my paper, Vitambara, Jeff, and Bill. And because Bill had forgotten to look for postdocs in his main keynote, I made sure to add this. He is looking for postdocs. He's sitting right there, so feel free to swarm him. Yeah, also ISMB for giving me money to come here.
Thanks a lot for this insightful talk.
Yeah.
Questions? Please.
Yeah, first is partly more of a comment. So our team has been one of those lines you showed for the Dream Challenge. And in the Dream Challenge, we basically had the hybrid setting you mentioned.
Yeah.
So comparing that to an average predictor that sees the same assays in that cell type is, from my perspective, kind of unfair. Mm-hmm. Because this is, none of the approaches could see those data when building the models.
Yeah, so the results that I showed here weren't in the hybrid setting, they were in the cross-cell type setting. So I was using a chromosome that the participants trained on.
For evaluation as well. Mm-hmm. So this hasn't been the challenge result.
This is not the same as the challenge setting.
Okay, that explains it. The other comment I have, or a question more, you said that if you put instead of the DNA sequence just random numbers, the model could also memorize that. But the epigenetic assays you use, those could just be random numbers in some order. So couldn't the model also memorize those random numbers from the epigenetics assay?
Yeah, also I want to jump back to the previous question. I chose CTCF there because it was an extreme example of what was going on. Many of the participants beat the average activity in many of the settings. So I'm not trying to say that the people there, that their models are not great or anything. There is simply one example here where things can get really bad. And so that's what you should take into account. As for your next question about epigenetics, the fact here is that it has to be the same across all cell types. And so the epigenetic activity does differ from one cell type to the next. And that's why the models that were built using the epigenetic activity don't suffer from this pitfall. That it has to be features that are the same from one cell type to the next. Does that make sense?
Yeah, makes sense. Thank you.
Yeah.
Okay, we have time for one quick question.
So very quick question. You basically mentioned that it's really easy to do things badly. Is there any rules to make things not badly? Because I imagine you use this in avocado. It looks a lot like the scheme of avocado that Will Noble presented. So how did you manage to not be horrible? Or were you horrible in avocado?
I think that when you're doing things in the first thing is when deciding on the setting to use, think about the setting that people would use your tool. If people would use your tool on an entirely new cell type, you have to use the cross cell type setting. If what you're doing is just trying to understand biology, it's not so necessary to do that. As for doing things correctly, I think that the clearest thing you can do is just compare against the average activity baseline. If you're beating it, you don't have to worry. And many tools that are in the cross-cell type setting may already be beating it. If you aren't beating it, then it may be worth trying to understand why. There was a talk by one of the Anshul Kandaji students. His name was Surag Nar. He gave a talk a few days ago where what they found is simply adding in the average activity across the training set to their model significantly boosted performance past it. And so if you include it directly in your model, now you're just boosting on that. So that might be a way to be able to improve performance. But to be clear, just compare against the average activity baseline. If you're beating it, you're good.
Thanks.
Thanks a lot for your talk.